Welcome to another episode of the Neuroanatomy Series run by NANSIG. My name is Ashwin Venkatesh and I'm an F1 doing a neuroscience-themed AFP in London, and in this episode we're going to take a deep dive into the special senses. In this video, we aim to cover the core knowledge you'll need to understand the visual, auditory and olfactory neuroanatomical pathways and their clinical significance, and I'll conclude with some suggestions for further reading for your interest. So first off, let's start by getting to grips with the visual pathway, which is arguably the most well characterized. We start by first appreciating what I like to call the inverted relationship between the retina and the visual field. Light coming from above strikes the inferior retina, whilst light coming from below strikes the superior retina. This means that the inferior retina subserves the superior visual field, whilst the superior retina subserves the inferior visual field. It is also important to appreciate that light coming from the side strikes the nasal retina of the ipsilateral eye and the temporal retina of the contralateral eye. Take a moment just to digest these facts. So once phototransduction has taken place in the superficial photoreceptor layer of the retina, the electrical signal first travels deeper to the ganglion cell layer, whose cells unite to form the optic nerve that exits the eye via the optic canal. The nasal fibers of the optic nerves then undergo a decussation at the so-called optic chiasm and join the temporal fibers of their counterparts to form the optic tract that passes through to the lateral geniculate nucleus of the thalamus. By this stage, some important fibers have already been given off. Firstly, fibers to the hypothalamus that regulate circadian rhythms, and secondly, fibers to the midbrain superior colliculus, which is involved in the pupillary light reflex. Returning to the thalamus, two projections, known as Meyer's loop and Baum's loop, constitute the optic radiations. The former carries fibers corresponding to the inferior retina, and thus the superior visual field, via the temporal lobe, whilst the latter carries fibers corresponding to the superior retina, and thus the inferior visual field, via the parietal lobe. Finally, the visual information terminates in the primary visual cortex, located in the calcarine sulcus of the occipital lobe, before higher-order visual processing takes place to form the visual percept. Hopefully by now you have a working mental model of the visual pathway. If not, please rewind and review the information another time before we proceed to the visual field defects. Visual field defects are clinically important neuro-ophthalmological phenomena to be aware of, since the pattern of the field defect maps closely to the anatomical territory that is pathologically affected. Using the underlying anatomical principles that we've discussed, try and work out first what would happen to the patient's vision if each of the lesions marked A through K were to occur. The most obvious pattern of loss is a monocular blindness, which is marked B. This reflects a loss of all the sensory information communicated by the eye. The optic nerve, therefore, is the prime target in this case and can be affected by vascular occlusion, multiple sclerosis, or retinal detachment. More subtle, however, are the central scotomas, labelled A, which reflects disease at the level of the macula where the cone cells that subserve the central visual field are clustered. Moving on to C, the lesion affecting the optic chiasm, since the nasal fibers are affected, which subserve the temporal visual field, we get a bitemporal hemianopia, characteristically caused by pituitary tumours or craniopharyngiomas in children. Then we have lesions affecting the optic tract, and these cause a contralateral homonymous hemianopia. What this means is that information from the opposite visual hemifield is lost in both eyes, which makes sense, since this affects the nasal fibers of the contralateral eye and the temporal fibers of the ipsilateral eye. Moving on to lesions that affect the optic radiation, remember the two loops, Meyer's and Baum's, that traverse the temporal and parietal lobes respectively. If the temporal lobe is affected, then you would see a contralateral superior quadrantanopia, whilst parietal lesions cause a contralateral inferior quadrantanopia. This is commonly remembered by the mnemonic PITS, parietal inferior, temporal superior. Lastly, in cases of posterior cerebral artery strokes, it is possible to get a contralateral homonymous hemianopia with macular sparing that is startlingly similar to the defect that we've seen previously caused by optic tract lesions, but subtly different. The reason for this is that whilst the posterior cerebral arteries supply much of the visual cortex, The macular fibers specifically are supplied by a collateral circulation between the middle and posterior cerebral arteries. This affords an added inbuilt redundancy that protects the salient visual information coming from the macula. And that's about all you'll need to know for visual field defects. Let's now move on to discuss the auditory pathway. Sound vibrations are captured by the pinna and transmitted down the external auditory meatus to the tympanic membrane, whose movement causes vibration of the middle ear ossicular chain, terminating in the contact made by the bony stapes and the oval window through to the fluid-filled inner ear. There, hair cells situated on the basilar membrane of the cochlear duct carry out mechanotransduction. Notably, a so-called tonotopic map is elaborated along the basilar membrane, whereby high-frequency sound waves cause maximum displacement of the basilar membrane and stimulation of hair cells at the base of the cochlea, whereas low-frequency sounds maximally stimulate hair cells at the apex of the cochlea. This aids in frequency or pitch discrimination. These hair cells are innervated by peripheral axons deriving from the spiral ganglion bipolar cells, whose central axon counterparts from these bipolar cells form the cochlear part of the 8th cranial nerve. All of the axons in the cochlear part of the 8th nerve enter the pontomedullary junction and synapse in the ventral and dorsal cochlear nuclei. Axons of cells in the ventral cochlear nuclei bilaterally innervate the superior olivary nuclei in the pons. These are the first auditory nuclei to receive binaural input, and use this binaural input to localize sound sources. The lateral lemniscus carries auditory input from the cochlear nuclei and the superior olivary nuclei to the inferior colliculus in the midbrain. Each lateral lemniscus carries information derived from both ears. However, input from the contralateral ear tends to predominate. The inferior colliculus sends auditory information to the medial geniculate body of the thalamus. From there, the auditory radiation projects to the primary auditory cortex, located on the posterior portion of the transverse temporal gyrus. The adjacent auditory association area makes connections with other parts of the cortex, most famously including Wernicke's area, which is the cortical area for speech and language comprehension. Hearing loss is commonly characterized as conductive or sensory neural. The former reflects disruption of passage of sound waves through external or middle ear, such as through waxy obstruction, otosclerosis, or otitis media. The latter reflects damage to the cochlea, the 8th nerve, or the central auditory connections, and 2 clinical examinations, known as Weber's and Rinne's tests, can be helpful to differentiate the two. Next, we have the olfactory system. The flow of information starts at the olfactory epithelium, where odorants dissolve in the mucosa and interact with ciliary receptors, which triggers a G protein cascade that depolarizes the olfactory nerve fibers. These axons pass through the cribriform plate to the olfactory bulb. There they excite mitral cells and smaller tufted cells in the olfactory glomeruli. The pattern of glomeruli activated in each bulb is distinct between different odorants. Odor-tuned mitral cells then transmit information via the lateral olfactory tract to regions of olfactory cortex, as shown in the figure. Interestingly, it is thought that COVID-19 causes anosmia due to effects on the supporting sustentacular cells in the olfactory epithelium, as well as the basal cells that regenerate the epithelium, rather than direct effects on the sensory neurons themselves. This might explain why COVID-related anosmia is more likely to be temporary than permanent. Note that many of the sensory pathways that we have discussed have featured a synapse within a thalamic nucleus, which serves as a hub where the information is sorted, integrated and analysed before being sent further to the cerebral cortex. This fact makes the thalamus a so-called gateway to the cerebral cortex for all sensory modalities besides olfaction, and also limbic and motor functions. Lastly, note that the cortex, including the sensory cortex, was historically organized by the German anatomist BRODMANN into cyto-architecturally distinct regions, the important ones of which are highlighted in the table. So to summarize, we have focused on the visual pathways and visual field defects, the auditory pathway, and the olfactory system, along with their clinical correlates. For those of you who are interested, I've included some extra reading here, and put forward a few papers exploring the association between impairment of the special senses and dementia. In a broad sense, it appears that advanced sensory processing constitutes a complex cognitive function that is directly vulnerable to the same pathophysiological processes that cause dementia, so it's perhaps unsurprising that the two are related. But could there be an element of causality? Here are a few acknowledgements and sources. Thank you for watching this NANSIG neuroanatomy video. Please do tune in to the other videos created as part of this educational series. Thanks again.